Welcome to the TOEFL Listening section. In this section, you will listen to an academic lecture and then answer questions about what you heard. You may take notes while you listen. You will have time to review your notes before answering the questions. Listen to part of a lecture in a university class. You will hear the lecture only once, so listen carefully. After the lecture, you will answer some questions about what you heard. Some questions ask about the main ideas and supporting details. Other questions ask you to make inferences or connections between ideas. Answer each question based on what is stated or implied in the lecture.
Good morning, class. Today we will explore bacterial resistance evolution and how resistance mechanisms arise and spread in microbial populations. We will examine genetic mechanisms such as chromosomal mutation and mobile genetic elements, and we will look at real examples like methicillin-resistant Staphylococcus aureus and carbapenem-resistant Klebsiella. First, consider mutation-driven resistance. A single base change in a target gene can reduce antibiotic binding and lead to resistance. For example, point mutations in the GIRA gene confer resistance to fluoroquinolones. An important early experiment was the Luria and Delbruck fluctuation test. This experiment showed that resistance mutations arise spontaneously before exposure to antibiotic and are not induced by the drug. Beyond mutation, we must emphasize horizontal gene transfer as a major driver of rapid resistance evolution. Horizontal gene transfer moves genes between bacteria by conjugation, transformation, or transduction. Plasmids often carry multiple resistance genes. For instance, extended-spectrum beta-lactamases are frequently plasmid-encoded and can spread between E. coli and Klebsiella in hospitals. Transposons and integrons capture and shuffle resistance cassettes so that one mobile element can assemble several resistance determinants on the same plasmid. I emphasize the role of horizontal gene transfer because it explains rapid dissemination of resistance genes across species. Mechanistic categories are useful to organize ideas. We see enzyme-mediated drug inactivation, such as beta-lactamases. We see target modification, like rRNA methylation that prevents macrolide binding. And we see reduced intracellular concentration through efflux pumps and reduced permeability. An important concept is selective pressure. When antibiotics are used extensively in medicine or agriculture, susceptible bacteria are killed and resistant ones expand. The selection amplifies rare variants, whether they arise by mutation or arrive by gene transfer. There are real-world case studies that illustrate these principles. In one hospital outbreak, a plasmid encoding a carbapenemase moved through several Enterobacteriales species, leading to a multi-species cluster of carbapenem-resistant infections. Agricultural use of tetracyclines has been implicated in selection for tetracycline resistance genes in farm bacteria. Those genes can move into human pathogens via environmental routes or food chains in a One Health context. One challenge is the fitness cost associated with resistance. Some mutations reduce growth rate in the absence of antibiotic. However, bacteria can acquire compensatory mutations that restore fitness while retaining resistance. Phylogenetic and genomic analyses allow us to trace clonal expansion and horizontal gene transfer events. Sequencing of MRSA lineages, or ESBL, producing E. coli, shows how both clonal spread and plasmid transfer contribute to epidemiology. From a public health perspective, interventions include antibiotic stewardship to reduce selective pressure, infection control to limit spread, and surveillance to detect emerging resistance early. Finally, research into novel strategies seeks to inhibit specific resistance mechanisms. For example, beta-lactamase inhibitors restore activity of some beta-lactam antibiotics, and research into efflux pump inhibitors continues. To summarize bacterial resistance, evolution is driven by mutation and horizontal gene transfer. It is shaped by selective pressures and modulated by fitness costs and compensatory changes. Understanding these processes is essential for controlling resistance. That concludes the lecture on bacterial resistance evolution, and I will take questions now.
Listen again to part of the lecture. Then answer the question.
I emphasize the role of horizontal gene transfer because it explains rapid dissemination of resistance genes across species.
This concludes the listening section. For more TOEFL practice tests and study materials, visit spinta.org.